Hello. What's up, Samir? How you doing?
How you doing, Sid? Good to see you.
Good to see you too. How's your break been?
Doing all right. How about you?
It's been decent. It's just gone by super fast.
I feel like way too quick.
Yeah. What's up, Cam? How are you doing?
Hey, how are you?
I'm doing good.
Break's been good.
Yeah.
Too fast, I agree.
Yeah. It's probably been the fastest break I've had at VCU.
Hello.
Hey, how are you doing?
I'm good. How about you?
I'm doing good. Let's wait a few more minutes to get started. It's, it's I think we can go ahead and get started. All right. So welcome to today's MCAT Max session. So if you haven't been here before, usually what we do is we go over a background topic, and then we do a practice passage. So first we start with the background PowerPoint. So this is the PowerPoint. So today we're going to be looking at a passage about class one cytokine receptors. All right. And before we start, let's do a little icebreaker so everyone gets to know each other. So if we could go around, just say your name, and then the icebreaker for today is, what's the favorite holiday gift you've received? Yeah. So whoever wants to go first can go.
I can go first.
Hmm.
My name is Samir Nader. I'm a student at VCU. Um, and my favorite holiday gift that I received was this egg boiler steamer thing. You put like six eggs in it, and it like perfectly cooks the eggs like a perfect temperature.
Oh,
So
nice.
looking forward to using that.
Yeah.
I'm Rana.
Hmm.
I'm also a student at VCU. I didn't really get anything. Um, we just kind of chilled for like the holiday season, but I guess I'm in California, so I'll count that. My name is Cameron. I go to VCU, and the favorite holiday gift I received was I just got a bunch of materials to begin to like sew and crochet. So I guess that was a pretty good gift.
Nice.
I'm Andy. I'm also from VCU, and I also kind of didn't really get any gifts, but it's been nice spending time with family for break.
I'm Mary, and my favorite holiday gift would be a snow globe that I got.
Oh, nice. All right. So we'll go ahead and get started. So today we're going to be looking so the name of the passage is called the class one cytokine receptor. And so basically what that is a protein. So we're going to start with some basics about proteins. So proteins have these special regions called domains. Can everyone see my mouse on the screen? All right. So proteins have special regions called domains that have like special purposes. So here we have a 3D structure of a protein. And you see there's different colors. So there's blue, green, purple. And so these are different domains of a protein. So these are specialized regions of a protein that have different functions. All right. So now that we have the idea of a domain down, you have to know that proteins, just like animals evolve from one another, organisms evolve, proteins also evolve. So some proteins are more closely related to other proteins, are more closely related to each other than other proteins. So we actually have what's called a protein family. So class 1 cytokine receptors is just another term for it's just an example of a protein family. So these proteins share similar structural characteristics, and they have these similar structural characteristics because they have a similar recent common ancestor. So they evolved along a similar path. So these proteins reside within a family. And so another, just like we talked about domains, another word is motif, which is a type of structural pattern in a protein. Yeah. So, and then also one of the major types of a protein is a receptor. And so receptors respond to signals. And so you can have what's called a homomeric receptor, which is it only associates receptors of the same type, or you can have a heteromeric receptor, which associates with receptors of different types. So if someone could explain the concept of like what is a domain? Anyone has a good grasp on that?
I have a question first.
Yeah.
What do you mean at the end by the homomeric receptors associate with receptors of the same type? What do you mean by the association there?
So association is like, for example, like if you form a dimer, so you have two receptors of the same type coming together to form like a dimer, or yeah. So if you had a like dimer receptor, it would be two subunits. They would be the exact same. But if it was a heteromeric receptor, it could be different. So, and you'll see an example of that too. Does anyone have any questions on that? All right. Okay, and so why do we care about receptors and protein families? It's because they're involved in signaling pathways in cells. So on the biology section of the MCAT, they like to ask about cell signaling pathways and receptors. So that's why it's important to go over them and learn how to answer questions about them. So one of the big ones, which is the one that's covered in this passage, is the JAK-STAT pathway. So those words seem kind of foreign. Well, the thing to know is they're just proteins. So JAK is just a protein. STAT is just a protein. They both bind to this receptor. So you see these two purple like sticks here. So those are cytokine receptors, right? And so basically JAK2 comes and binds to this class 1 cytokine receptor. So this class 1 cytokine receptor, this purple stick, it's called a single pass transmembrane receptor. So based on the name single pass transmembrane, what does that tell you about the protein?
Goes through the membrane one time.
Yeah, exactly. So you can see right here in the picture, the purple stick is it's going through the membrane one time. And so this here is an example of a dimer of receptors. So remember how we were talking about homomeric versus heteromeric? This would be homomeric because each subunit of the dimer is the exact same. All right. So basically in the JAK2 STAT pathway, what happens is we have an outside signal, which is our ligand, binds to the receptor. When it binds to the receptor, it leads to a change in the conformation of the receptor, and that leads to the JAK2 protein being activated. When the JAK2 protein gets activated, the tyrosine residues on the receptor get phosphorylated. When these things get phosphorylated, STAT3 can dock on the membrane and get phosphorylated. And STAT3 is kind of like the end product of this pathway. It's the most important part because it goes into the nucleus and changes gene expression. So it acts as a transcription factor. So could someone tell me what a transcription factor is?
I'm pretty sure a transcription factor binds the DNA and
Hmm.
begins DNA transcription.
Yeah. Yeah. So yeah, so it can help with beginning it or it can alter it, but yeah, just basically affects gene transcription. All right. Yeah, and so remember how we said that cytokine receptors are family, meaning there's a ton of receptors that are very similar to each other. So actually, growth hormone receptor is part of this class 1 cytokine receptor family. So it naturally exists as a dimer like this. All right. And these JAK2 and STAT proteins associate with it. So does anyone want to like take a shot at trying to like describe this pathway just to see if you got a good understanding?
I think I could.
Yeah, sure.
So this JAK-STAT pathway, the protein there exists as a dimer, or the receptor exists as a dimer made up of two single pass transmembrane pieces. JAK2 associates with this dimer, and once a ligand binds to the receptor, JAK2 and the tyrosine residues are phosphorylated. Once they're phosphorylated, the STAT protein can bind and also become phosphorylated. And once that is activated by phosphorylation, it can go into the nucleus and change gene expression.
Yeah, that sounds perfect. Good job. All right.
Thank you.
Yeah. Yeah, so that's the whole pathway right there. So that's kind of like the most complex part of it. But you just gotta, it's always good to just like simplify it in your head, just like because they will have like a lot of weird names like JAK2, STAT, but as long as you know they're just proteins, you have a receptor and they're binding to one another, then you're good. So let's go on to the next slide. So okay, so a weird kind of like naming convention that I kind of get tripped up about is that the word receptor can be used to refer to the individual like receptors themselves or the dimer itself. So this whole thing together can be called growth hormone receptor, or you can call the individual subunit growth hormone receptor. So that's just something to be aware of, so you don't get tripped up about it. Okay, and so remember how we're talking about proteins? So another important protein in your body is called cytochrome P450. So cytochrome P450 is important because it has this thing called a prosthetic heme group. Can anyone tell me what a prosthetic heme group does? Like, why is that important? Well, so basically what a prosthetic heme group does is it allows the protein to accept electrons. So you see this iron right here in the center of the heme group? Iron can accept electrons, right? So if something can accept electrons, is it going to act as an oxidizing agent or a reducing agent?
Reducing.
So it would be oxidizing because if it's taking electrons from something, then it's oxidizing it. Right? Because so basically let's say you have a random compound, right? And then you have cytochrome P450. This protein is going to come to the random compound and it's going to pull electrons from it, right? Because it has this prosthetic heme group. So a prosthetic heme group, it's bound to the protein and it's crucial. Remember how we talked about prosthetic groups in the past? They need to be on the protein for the protein to work. So this prosthetic heme group, because it has an iron atom at the center, and iron likes to get reduced, it's going to pull electrons from whatever the protein's interacting with, and it's going to oxidize that substrate. So this is really important in your body, especially in your liver, for like detoxifying drugs, for like oxidizing the drugs that come into your body so they can be cleared by your liver and eventually your kidney. And then it also helps with making certain like hormones and stuff like that. So the important thing to know is cytochrome P450. And like this is one of like those like trivia facts that MCAT just likes to throw in there on like a lot of tests. That like what does cytochrome P450 do? And you just need to know it oxidizes compounds. So just a fact to commit to memory. Right? And it does it because of this heme group. Yeah, so it's also good to know what heme is for the MCAT because they asked about that. All right. Anyone have any questions so far? Nope. All right. Awesome. So now we're going to go over our usual concept of amino acids because they're really important through almost every section of the MCAT. So can anyone tell me what the aromatic amino acids are?
Tryptophan, tyrosine, phenylalanine.
Yeah.
And yeah.
That's it, right? Yeah. Yeah, so those are the aromatic amino acids. So a lot of times in the MCAT, they'll like ask you a question. They'll be like, what amino acid would you expect to find in this protein or structure? But what they really want you to do is they want you to go back and look at the passage because they'll usually give you the answer straight up. But they have to make you look a little bit in the passage. So you'll see an example of that in this passage. So it's really important when like the passage mentions about like certain types of amino acids being in certain structures, you want to kind of keep that in the back of your mind. So you can easily go back when you're reading that question and get that information because usually the answer is right there in front of you.
Wait, would histidine also be aromatic?
Histidine, I don't think so. Yeah, I'm trying to think too. Yeah, I'm gonna say no for now. I'd have to look into it more, but I don't think so.
Okay.
Yeah. Okay, and then also, I think we talked about this before, but if you have a transmembrane region of a protein, what is the area around that transmembrane region going to be like? Is it going to be polar or non-polar? So this is cutting through the cell membrane. Is it going to be non-polar or polar?
Non-polar.
Yeah, it could be non-polar. So if you have a transmembrane region and a non-polar region, remember like likes to mix with like. So you don't want polar residues in there. You want non-polar residues. So you want non-polar residues in transmembrane regions of the protein. All right. Does that make sense to everyone? All right. And as always, just like make sure you know every amino acid, like all its properties, because they'll 100% ask about it on the test. So it's just good to know. All right. Anyone have any questions before we go to the passage? All right. Let me share my screen. Awesome. All right. So if someone could read, I guess the first sentence.
I can read it.
All right.
Just the first sentence.
Yeah. You can read the first paragraph too if you want.
Okay. Assistant. Substantial number of key class 1 cytokine receptors utilize Janus kinase 2 JAK2 for signaling. Class 1 cytokine receptor structures are single-pass transmembrane receptors with an extracellular domain (ECD) structure containing at least one cytokine receptor homology domain, which possesses two fibronectin 3-like domains, each with an immunoglobulin-like beta sandwich domain with 7 strands and 2 layers. The membrane proximal domain contains a WS-XWS motif, YGEFS in the GH receptor, creating an aromatic stack.
All right. So remember how we talked about class 1 cytokine receptors? And we talked about how they're a family. So this is giving us all the family of receptors, and it's giving us each receptor type right here. And so within each receptor, so each like stick basically, they have different parts. So these different parts are the domains. So in the passage here, it says that each cytokine receptor has an extracellular domain. So if the extracellular domain, where is it going to be in this diagram? Like which side of the membrane is going to be on?
The top side.
The top side, so the outside of the cell. So within each extracellular domain, it has at least one CRH domain. So this here on the diagram, it defines what a CRH domain is here, right? They all have this common, I guess, like dark gray, white. So you can see every one of these has a dark gray, white region. All right. So we got through that. And so within each CRH domain, it says there's two fibronectin 3 like domains. So it tells us here in the key that a fibronectin 3 domain looks like this. So this is, you can tell this is kind of like with the like individual subunits of this receptor, cytokine receptor homology domain are kind of like this. So they're fibronectin like, all right. And then it tells us each of these ones, so like within this, there's an immunoglobulin like beta sandwich domain. So that's not pictured on the diagram, but this is telling you it exists, right? And then here it says the membrane proximal domain contains the WS motif. So we're talking about the CRH domain on the outside, right? And it says membrane proximal. So based on the word membrane proximal, which domain is talking about? It's talking about the white one or the dark gray one?
Dark gray.
Yeah, exactly. Because it's the one domain closest to the membrane. And you can see it does have the WS times WS motif. See, it says it has it on the key right here. All right. So does everyone have a decent understanding of the receptor structure? Okay, good. All right. So here it says domain structures depicted for the homomeric signaling receptors and examples of heteromeric receptors. So remember how we said heteromeric receptors can interact with other receptor types? So here you see these receptor types can interact with this gamma C, which is different, right? Or it can interact with this, what is this called? CNTFR alpha, or no, it's GP130. So these three interact with GP130, which is different from this. So that's why it's heteromeric. All right. Okay, and now if someone could read this second sentence. That's just a sentence. Yes.
I could do it.
Yeah, sure.
The growth hormone receptor is a dimer with just one CRH domain. Is the simplest of the class 1 cytokine receptor.
Yeah, okay, so it's like a typo in the sentence. It doesn't really make sense, but yeah, so this is the diagram of the growth hormone receptor, and you can see here it is a dimer. So we have two growth hormone receptors paired together, all right? And it has one CRH domain, all right? All right, and then if someone could read the last paragraph.
I got it. A second major feature of this receptor class is the proline-rich box 1 motif, which together with the less conserved distal box 2 sequence of aromatic and acidic residues serves to bind the JAK2 close to the inner cell membrane. These receptors primarily use JAK2 to activate the STAT regulators of gene transcription. JAK2 activation is followed by tyrosine phosphorylation within the receptor cytoplasmic domain, which generates docking sites for SH2 domains containing proteins such as STAT3 or STAT9, so that the docked protein is phosphorylated and activated.
Awesome. Okay. So it mentions the proline-rich box 1 motif. So where is that in the diagram? Does anyone spot it?
Yeah, see, it's on the intracellular side, proximal.
Yeah, exactly. So remember in the diagram, the PowerPoint, we talked about how JAK2 binds to the inside, the part on the inside. So that's where it would bind, box 1 and also box 2, which isn't shown on here. All right. And so it talks about, like we talked about, it uses JAK2 to activate STAT3 because when JAK2 is activated, this tyrosine residues on the receptor get phosphorylated. So that stat can dock and get phosphorylated, right? So that's that. So anyone have any questions on the passage? Awesome. And now we can attack the question. So it says here, which amino acids are likely to be found in the WS times WS motif? So it mentions the WS times WS motif here. So it says the membrane proximal domain contains the WS times WS motif, creating an aromatic stack. So the motif creates an aromatic stack. So what does that kind of tell you? What type of residue could be in there?
It would need to be aromatic.
Right. So based on the options you see on the screen, what is the answer?
D.
Yep, D. All right, awesome. Because it's tryptophan, so that is aromatic. That is the correct answer. All right. Question 2. During X-ray crystallography, so X-ray crystallography just basically lets you see the 3D structure of a protein. Which amino acid would most likely be found in the 270 position of a growth hormone receptor? So it's talking about growth hormone receptor, and here they give us a diagram of growth hormone receptor, and they happen to have numbers here. So we need to find which region number 270 would be in. So which region is going to be in?
I think it would be the extracellular portion, but towards the toward right on the membrane, but it's still extracellular.
Let's see. So which one? What's the name on the diagram? So based on the number ranges here.
Oh, 270. It would be intracellular, sorry, and it would be the membrane.
Yeah, so it'd be the transmembrane domains because this is 263 to 288, right? So 270 would be in that. Does it make sense to everyone? So it's going to be a transmembrane domain because 270 is within 263 to 288, right? So if we know it's in the transmembrane domain, what does that tell you about the residue? What property does it have to have?
It needs to be nonpolar.
Right. And so based on the options there, what is the nonpolar residue?
I think it's B.
Yep. I think B is the correct answer. Everyone else? All right. Yep, B is the correct answer because glutamine is polar, arginine is positively charged, lysine is also positively charged. So we don't want charges in the membrane. So awesome. Okay, question 3. Growth hormone upregulates the activity of cytochrome P450. What effect does growth hormone binding to P450 enzymes have on substrates? So what did we say cytochrome P450 does?
It's an oxidizing agent.
Right. So if you have upregulated activity, what does that mean?
It's gonna oxidize more things.
Right. So the correct answer is
A.
right. Yeah.
Is cytochrome P450 mentioned anywhere in the passage, or is it just like assumed that you need to know?
Nah, so yeah, that's just a fact you got to know.
Oh wow. All
Yeah.
right.
So that's why, yeah, it's just kind of like a rare fact they like to throw out there. Test
And that's biochem, correct?
Yeah, biochem, yeah.
Okay, interesting.
Which I didn't even learn that in biochem, so
Can you re-go through the logic of that question again one more time? Sorry.
Yeah, so cytochrome P450, you need to memorize for the MCAT. It oxidizes compounds because it has a heme group in it. The heme group has iron. Iron accepts electrons. So because it has a heme group, this cytochrome P450 can accept electrons and oxidize things. And so if growth hormone upregulates the activity of cytochrome P450, you have more oxidation happening. And so that's why A is the correct answer.
Do you know how
Mhm.
the how growth hormone upregulates cytochrome P450? Like what the
I don't know. I think it's just the question is telling you that that's just a fact. Like, so you just have to assume what the question tells you.
Okay.
Yeah.
And here, just a substrate just means any compound that comes in contact with
Yeah,
the
exactly. So any compound
with the thing.
interaction. Yeah.
Okay. And wait, what is exactly cytokine P450? Is it
So
a protein?
let me go back to the slide because it has
I know it has the heme group, but like, what's that like called? Like, what's that complex called? Like, it's like class, I guess.
so cytokine P450 itself is a protein.
It's just a
And
protein. Okay.
yeah, and so
All right.
it actually belongs to, so cytochromes tend to be a family of proteins. And all those proteins have heme groups in it, right?
Okay.
So those heme groups, so you might know like the electron transport chain, the mitochondria, there's cytochrome C, I think is it, yeah, cytochrome C. That thing carries electrons, and it carries electrons because it
Okay.
has this heme group. So the iron can always accept electrons and carry electrons. So, but in
Interesting.
cytochrome P450's case, it always accepts electrons. It always oxidizes stuff. So it's just a protein. But remember, we talked about prosthetic groups, which is it needs this heme group to function.
Okay.
Yeah.
So the main function of cytochromes is to electron transport.
Yeah, exactly. And cytochrome P450, it's always to oxidize. So
Okay. So to grab the
yeah,
electrons, basically.
exactly.
Okay.
Yep. Okay. So, according to the passage, binding of GH, the GHR cytokine class 1 protein will not result in which of the following. So let's go through this answer choice by answer choice. So the hormone is called growth hormone. So would we expect it to increase the cell growth? Right, we would, right? So we can eliminate that. So then it says binding of JAK2 to box 1 and box 3. So that sounds kind of specific. So let's look back at the passage. So here it says that JAK2 binds to the box 1 motif and the box 2 sequence in the signaling pathway. So do you see box 3 mentioned there? It's not, right? So okay, so we think maybe B might be the correct answer, but let's keep looking. Here it says increased tyrosine kinase activity. So tyrosine kinase phosphorylates to tyrosine residues. So if we have more of this pathway happening, remember we know that tyrosine gets phosphorylated in this pathway. So if we have greater activity of this pathway, we are pretty sure that we're gonna have more tyrosine kinase activity, right? So we can eliminate C. And so this pathway, you know, there's a lot of stuff getting phosphorylated. And to phosphorylate stuff, you need ATP. You need to use ATP. So if this pathway is more active, we need to have increased ATP usage. So
Wait, can you repeat the reasoning for D again?
yeah,
There's more ATP usage because why?
So we know things are getting phosphorylated in this pathway, right? The tyrosine receptor itself gets phosphorylated, STAT gets phosphorylated. And to phosphorylate something, you're adding a phosphoryl group. And so that phosphoryl group always comes from ATP. So if you're going to phosphorylate more stuff, you need more ATP. So you need increased ATP usage. And that makes sense to everyone?
Yeah.
All right.
And you said it's not C because in the GHR pathway, JAK2 is followed by tyrosine phosphorylation, and to phosphorylate you need a kinase.
Yeah, exactly.
Okay. All right. Makes sense.
Yeah. So B is the correct answer. And last question. A defining characteristic of STAT proteins is that they. All right. let's go through this choice by choice again. A, they form homodimers. So in the passage, it mentions stat proteins at the very end, but does it mention anything about whether they dimerize or anything like that? Do you see anything about that?
I think. Did you ask a question? I'm sorry.
Oh yeah, so yeah, I was just asking, did you see anything about dimerization when it comes to stat proteins in the
No.
passage? No, yeah. So they don't mention anything about stat forming dimers or anything like that. So we can eliminate that answer choice. They dimerize to other proteins. It doesn't mention anything, so it can't be a defining characteristic. Is like something we know stat proteins have this characteristic, right? That's what we identify with stat proteins. It doesn't mention anything about dimerization, so it can't be a defining characteristic. We know stat is it's a transcription factor, right? So if you're a transcription factor, the first thing you think of transcription factor is binding to DNA. So that is considered a defining characteristic. So defining is like a main feature. So that's pretty obviously can bind to DNA. And that D is tricky. They're like just trying to trick you right there. That it's present within the nucleus of the cell, but it's not a defining characteristic because this can exist in the cytoplasm. Remember, it has to go bind to the receptor, or it can exist in the nucleus. So defining characteristic would mean it's always in the nucleus. So it's not always in the nucleus. So that's not the defining characteristic. So the best answer would be C. It can bind to DNA because it's a transcription factor.
In the passage, also, I think that the important sentence was these receptors primarily use JAK2 to activate the stat regulators of gene transcription.
Yeah,
It kind of
exactly.
tells you.
Yeah. So yeah, it tells you that in the passage too. Yep. So C is the correct answer. I believe that's the end of the passage. Does anyone have any questions about the passage or the PowerPoint? No?
Not about
Oh.
the passage of the PowerPoint, no.
Okay. Oh yeah, go ahead.
But I have a question about so we have the cytochromes, the class of proteins that their job is to transport electrons.
Yeah.
Are there other classes of proteins that transport protons?
Oh, electrons?
That proton, like, oh wait, I guess stuff like
Yes.
NAD.
Yeah. So those are all those are like that they themselves are not proteins, but they can be bound to proteins. So NAD plus, NADH, those are electron carriers. So usually you think of those in metabolism. So in glycolysis, basically, and Krebs cycle, you make NADH, FADH2. So you're taking the electron, you're basically oxidizing. That's why they call it glucose oxidation, because you're pulling the electrons from glucose and you're creating electron carriers like NADH and FADH2, right? Then those electrons travel down the electron transport chain and reduce oxygen to water. And basically then you pump protons, create a gradient, and then you make ATP. So those are yeah, another example of electron carriers. And you actually have cytochrome C. Let me see. Do you see this picture right here? Do you guys see a picture?
Yeah,
All right. So you see cytochrome C is actually here. So it takes electrons from complex 3, carries it onwards. So yeah. And you can see NADH, FADH here, they get oxidized. So they're giving their electrons to the chain. So that was something you need to study more in detail. But yeah, so cytochrome P450 is an electron carrier. There's a lot of different electron carriers out there. So yeah.
thank you.
Yeah, no problem. Anyone have any other questions or good? All right. Well, great session today. Everyone's free to go. So have a good day.
You too. Thank you, Sid.
Yeah, no
Thank
problem.
You said, bye.
No problem.